Hello children, good evening. Welcome to AIMS India online classes, Biology, Cell. Today we are going to discuss about the cell, its structure and functions. So let us recall the previous knowledge. You already know that the discovery of cell and the shape and size of the cell, unicellular and multicellular organisms. So let us recall some of the activities which we have studied previously. You have learnt about the cells and their structure. You have also learnt about discovery of cell, its sizes and shapes, unicellular and multicellular organisms. Primarily, cells are studied under the optical microscope, simple microscope. The cell under compound microscope shows different structures or organelles: cell wall, cytoplasm, nucleus, chloroplast, mitochondria, etc. So this is an optical microscope, normal microscope, with which or a compound microscope it is, with which we can study the already prepared slides. When you observe under this microscope, it can magnify the size of that particular structure up to some 400 times, like that. So we can clearly see these structures under this compound microscope. But exactly in order to study the structure and function of cells, you need an electron microscope. An electron microscope is necessary. So let us proceed. All the organelles shown in the typical plant or animal cell will not exist in every cell. Means whatever the organelles which are present in plant cell or animal cell, they are not present in all the cells. The meaning is, for example, chloroplast present only in the cells of green plants and parts like leaf, tender stem. Tender means green stem. But the chloroplasts are not present in other parts like roots. That is the meaning. The organelles that feature in most of the cells are included in a typical cell model. A typical cell provides a way to study cells. This is a typical cell. It is a model which represents the actual cell. The organelles like mitochondria, nucleus, ribosomes, vacuoles, chloroplast, cytoplasm, cell wall, cell membrane. So these are all the organelles we can see in a typical cell or a prepared cell. The organelles that feature in most of the cells are included in the model. So the typical cell provides a way to study the cells. So what common features we find in a plant cell and animal cell? Let us see. The difference between plant and animal cells. The left side represents the plant cell, which is in rectangular shape, and the oval shape, that is animal cell. Okay. So animal cell and plant cell show some similarities. Both animal cell and a plant cell have a nucleus, cytoplasm, plasma membrane, Golgi apparatus, mitochondria, endoplasmic reticulum and ribosomes. So these are the common. So cell membrane, nucleus, nucleolus, smooth endoplasmic reticulum, rough endoplasmic reticulum, ribosomes, vacuoles, mitochondria, Golgi complex. These are all the common structures in both plant and animal cell. So let us see the differences now. If you see the shape of this plant cell and animal cell, plant cell have fixed shape, they possess fixed shape, but animal cells do not have fixed shape, they are of different shapes. Next, cell wall, a typical structure, cell wall. So this cell wall is present in plant cells, it is absent in animal cell. The plant cell wall is present, but in animal cell wall is absent, they don't have cell wall. Next, vacuoles. So vacuole is another important structure which is present in all plant cells, but in animal cells these vacuoles are absent. If present also in some unicellular organisms, these vacuoles are present. For example, amoeba or protozoans, amoeba, paramecium, euglena, you possess these vacuoles. These vacuoles, they are small in size. If they are present in animal cell, they are small in size, but whereas in animals it is small in size, whereas in plant cell they are large in size. Almost more than 50% of the volume of the plant cell is occupied by this vacuole. The size of the vacuole represents the age of the plant cell. A mature plant cell possesses a very big vacuole, even it pushes the nucleus aside. Okay, but whereas in animal cells the vacuoles are absent. If they are present also in unicellular organisms like amoeba, paramecium, they are small in size, which involves in the digestion of food vacuole. And another type of vacuole in the animals, contractile vacuole, they helps in the osmoregulation. Next, chloroplast. Chloroplast are the typical structures, they are present only in the plant cells. There are no chloroplast in the animal cells. Because of presence of chloroplast, plants perform autotrophic nutrition, animals heterotrophic nutrition, because no chloroplast. These are green in color, which contain chlorophyll pigment and helps in a biochemical reaction, photosynthesis. So plants have chloroplast, animals do not have chloroplast. Food storage, another very important difference. The storage, plants store the food material in the form of starch, but animals store food material in the form of glycogen. In plants it is stored in different parts like roots, fruits, stem. Okay, example, potato, starch is present in the stem part. Fruit, banana, starch is present in the fruit. Okay, in the roots also. So different parts of the plant store the food material in the form of a starch. But whereas in animals, the food material, that is the carbohydrate, excess of carbohydrate, stored in the form of glycogen in the muscles and in the liver. Next, centrioles. Centrioles are the structures which are present only in the animal cells, they are absent in the plant cell. So these centrioles are present in the structure called centrosome. At the time of cell division, these centrioles they reach the opposite poles of the nucleus and help in the process of cell division. Okay, but whereas plant cells do not have centrioles. So that's why, but at the time of cell division, the plant cells also produce centrioles. Centrioles are formed at the time of cell division only in the plant cells. The main difference between plant cell and animal cell is cell wall, present in the plant cell, absent in the animal cells, and the green color plastids, chloroplasts, are present in the plant cell, absent in the animal cell. Lysosomes are present in the animal cell, absent in the plant cells. Okay, children, let us proceed. You can compare the vacuoles of plant cell and animal cell here. So plant cell vacuole is large in size, animal cell vacuole is small in size. Let us proceed. Cell membrane. The cell membrane is an important structure which is common in both plant and animal. Almost all living cells possess an outer membrane called a cell membrane. In order to understand this cell membrane, let us do one activity, that is Rio Leaf. This is the plant. Rio plant is an cotton plant. Take a Rio Leaf. Take a Rio Leaf from a petri dish, or isolate a leaf from this plant, and fold the leaf and tear it along the lower side of the leaf. You can fold and tear the lower side of the leaf. Using a forceps, pull out two small segments of thin transparent layer from the lower epidermis of the Rio leaf. Okay, you can, with the help of your teacher, you can conduct this activity or experiment in the school laboratory. So place the epidermal peels on both glass slides and observe under microscope. When you observe under microscope, while observing, you can draw the picture like this. You can see the picture like this. So here, outer membrane, that is cell membrane. So before seeing it, you have to mount the slide. So our preparation of the slide, to prepare that, you have to prepare salt solution. Take 50 ml of water and dissolve 1 teaspoon of salt in it and stir it thoroughly, so that a solution is prepared. And after that, you can place the slide under that. So here you can see the shrunken cytoplasm. Shrunken cytoplasm you can see here. You can see the outer membrane. This is cuticle, outermost layer around the cell membrane. So this is the cell membrane actually, and it is the nucleus when you observe under the microscope. So you can compare your observation. The outer boundary colored area actually the cell membrane, which became separated from cell wall. This is the cell wall. This is the cell wall. So beneath the cell wall, cell membrane you can see. The cell membrane, it defines the shape and size of the cell, encloses the cytoplasm and protects the cell from the external environment. The cell membrane protects the cell from external environment. The internal environment of the cell is different from outside. So let us see more about the cell membrane. The structure of cell membrane is clear only through electron microscope. More clearly you can see this is the internal structure of cell membrane. When you observe the same cell membrane under electron microscope, you will see these structures. It is flexible and is made up of mainly lipids and proteins. The chemical composition of cell membrane or plasma membrane is lipids and proteins. The outer layer of cell that separates cytoplasm from the external environment. This outer side of the cell membrane, it separates the cytoplasm from external environment. This is also called a plasma membrane. The cell membrane is also called as a plasma membrane. Okay. So this cell membrane performs different functions. The cell membrane defines the shape and size of the cells. That is one of the function. The cell membrane also plays a crucial role in maintaining osmotic balance in the cells, and any substance entering or leaving the cell only through this membrane. That's why the cell membrane is also called as selectively permeable membrane. It is also called as semi-permeable membrane. It forms the connection between the cells. It forms the connection between the cells. So this is the structure once again. It is made up of proteins and lipids. The proteins are present, it acts like receptors, okay, and a bilayered membrane, lipid layer. Okay, these lipids are phospholipids, and it also contains a chain of carbohydrates on the surface of these proteins. A chain of carbohydrates are also present. So the cell recognizes, or cell recognition and communication is one of the important function of cell membrane, and it impart negative charge to the cell, repels other particles. So it also helps in intercellular attachment or adhesion. Okay, it acts as receptors. Then cell identity markers. So particularly these proteins, these brown colored structures, they are proteins. So glycoproteins, they are glycoproteins. That's why carbohydrates are attached to it. That's why they are called glycoproteins. And glycolipids. Okay, these lipids are glycolipids. The antibody processing takes place. That's the reason why whenever any foreign particle tries to enter into the cell, first these proteins, okay, the glycoprotein layer, glycolipids layer, it detect those antibodies. Okay. So once again, let us recall the functions. Any substance entering or leaving the cell can do so only through this cell membrane. So the uniqueness of the membrane is it only allows certain particles to pass through it. Unwanted particles it will reject. That's why it is called semi-permeable membrane, or it is also called selectively permeable membrane. The exchange of substances also takes place through this membrane, and hence it is a selectively permeable membrane. This characteristic of the membrane enables it to control the exchange of substances between the cell and its external environment. Okay, this characteristic feature helps the cell membrane to exchange of the substances between the cell and its external environment. Okay, we are going to learn in higher classes the movement of materials across the cell membrane. Okay. Okay, children, let us test ourselves how much we have learned. Okay, today we studied about the difference between plant cell and animal cell, and about the plasma membrane. So about the typical cell, a typical cell consists of different organelles like chloroplast, mitochondria, endoplasmic reticulum, nucleus. Okay, these are all present. So the structure of cell and its function, if you understood the structure, then you can understand the functions. In order to study the structure, whatever we are studying, that is electron microscopic view, not the ordinary microscopic view. Cell itself is the smallest unit of living organisms. So inside the cell, the organelles are very, very small, nano structures they are. Okay, so let us answer the questions, children. Which organelle is only found in a plant cell? Nucleus, chloroplast, mitochondria, cell membrane. Yes, very good. Chloroplast. Chloroplast is the structure present only in the plant cell, absent in the animal cell. Next question. I am the outermost layer of an animal cell. What am I? Cell wall, nucleus, cell membrane, cell skin. I am the outermost layer of the animal cell. What is the answer? Yes, cell membrane. Next question. What organelle is considered to be the gatekeeper for plant and animal cells? Cell wall, nucleus, cell membrane, cytoplasm. It is called the gatekeeper both in plant and animal cell here. Yes, cell membrane. Cell membrane, as we studied, that it is selectively permeable membrane. It allows only the substances which are required to pass through it. It does not give permission to every particle or every substance to pass through it. So that's why it acts like a gatekeeper. Next, which structure is the boundary between a living cell and its environment? Cell wall, nucleus, cell membrane, cytoplasm. Which acts as the boundary of the cell, living cells. Here again, cell membrane. The structures within the cell are known as the structures within the cell. Cells, vacuoles, mitochondria, organelles. What do you call those small structures within the cell? Organelles. Which structure is found in plant cells and not in animal cells? Cell wall, centriole, nucleus, cytoplasm. Which structure is found in plant cells and not found in animal cells among these organelles? Here cell wall, centriole, nucleus and cytoplasm is given. So the structure is found in the plant cells and not in the animal cells. Okay, here the answer wrong given. Cell wall. What is the right answer here, children? Cell wall. This is wrong. Which structure is found in plant cells and not in animal cells? So nucleus is found both, cytoplasm is also found in both plant cell and animal cell, but the cell wall is found in plant cell, not found in animal cell. Next, I am only found in plant cell. What am I? Here cell wall, mitochondria, chloroplast and cytoplasm is given. So analyze the question, read question twicely, otherwise we will commit the mistakes. I am only found in plant cells. What am I? So here cytoplasm found in both plant cell and animal cell, chloroplast found in plant cell, mitochondria found in both plant cell and animal cell, cell wall found in plant cell. So here only you have to underline. I am only found in plant cell. What am I? So here the organelle is also given. That is, what is this organelle? Yes, chloroplast. So the answer is chloroplast. So here if this diagram is not given, if this picture is not given, then your answer is cell wall and chloroplast. But here, as in the question this figure is mentioned, so the answer is C, chloroplast. Next, carrier proteins aid in movement during osmosis, diffusion, facilitated diffusion. Here also image based question. So here the carrier proteins, actually these are the proteins, special structures. The carrier proteins aid in the movement during. So what is this process? This process is called diffusion, but here carrier proteins are mentioned. Only what is this movement means? Diffusion you can say. But the carrier proteins aid in this special movement. This type of movement is called facilitated diffusion. Facilitated diffusion. The answer is C. Next, this image shows, the image is given here. This image shows endocytosis, exocytosis, osmosis, pinocytosis. So we need to recall the definitions. Endocytosis, pinocytosis, exocytosis, osmosis. Osmosis is the process where the plasma membrane, this is the plasma membrane. The plasma membrane is involved in all these processes. So this membrane, pinocytosis or phagocytosis, the membrane extends out and it will engulf the, it will surrounds the particles. That process is called as pinocytosis. Osmosis, the movement of particles takes place to and fro through the plasma membrane from high concentration to low concentration. When the two different concentrated solutions separated by this plasma membrane, the exchange of materials takes place. That is called osmosis. It is a physiological process, physical and physiological process. Whereas the pinocytosis, endocytosis, physical process. And this endocytosis, exocytosis and osmosis, these are physiological process. So here the plasma membrane is involving. So here this is representing as the particles are coming out. Okay, as the main function of plasma membrane is to maintain osmotic balance. Sometimes the particles will come out through the membrane and sometimes they go into the membrane. So here this process is called exocytosis, means from the cytosol the fluid is coming out. So exocytosis or exoosmosis. Next, the answer is B. Next question, what is not true about the cell membrane? It controls what enters and leaves the cell. It's a semi-permeable membrane. Phospholipid bilayer is present. Contains digestive enzymes. So here the correct answer is D. Okay, what is not true is asked here. So it is not true that cell membrane contain digestive enzymes. Actually, these digestive enzymes are present in the lysosomes, not in the cell membrane or plasma membrane. Next, what do the brown structures represent? Here also one image is given. What do the brown structures in this plasma membrane represent? What are these brown structures represent, children? Yes, it represents the channel of proteins. Channel of proteins. This green color things represent the carbohydrates. Chains of carbohydrates. So this entire structure represents the glycoproteins. Okay. So channel of proteins. Answer is C. Next question. The last question. The sodium potassium pumps requires energy. So it is a type of, which type of transport? There are two types of transports are there. One is passive transport, another one is active transport. So here the transport requires energy. The transport of sodium and potassium inside the cell, across the cell membrane, the transport is taking place. So it is utilizing energy in the form of ATP. So such transports are called active transport. Okay, active transport. These membranes, cell membranes, they are involving in almost all internal organs are covered by means of living membranes called plasma membrane or cell membrane. So it involves in the transport. It acts like a bridge through which the transport of substances takes place. That transport occurs in two ways. One is active transport, another one is passive transport. In active transport, energy is used in the form of ATP. Okay. Next question. What forms the channels and pumps in phospholipid bilayer? Here also proteins. What forms the channels and pumps? In the previous image only we have seen the channels of proteins, okay, and phospholipids bilayers, they involves the proteins. Proteins form the channels and pumps in the phospholipid bilayers. Okay, there are several models have been introduced to explain the structure of these plasma membranes. Okay, in your higher classes you are going to study like Robertson model, okay, the different models. Bilipid layer model, it is one of the model which we have seen in the today images. Okay, thank you for watching our video. Please subscribe our channel.